Welcome back to Chapter 12, Part 2. In this part, we're going to learn all about linked genes and crossing over. So, what we know about chromosomes, and that we've been simplifying so far, is that each chromosome has hundreds or thousands of genes on them, with the exception of the Y chromosome. Remember, that only has a few genes on it. But most chromosomes have hundreds or thousands. And what that means is that when you just think about passing down a chromosome, the information that is contained on that chromosome is going to be passed down as kind of a unit. I mean, there is some exception to that, and we're going to talk about those exceptions. But genes that are located on the same chromosome, we say tend to be inherited together, and the word that we use for that is linked. So if we look at this is chromosome number 1, and there are so many genes on chromosome 1, but all of these in general tend, if you've got the dominant version here and the recessive version here, your offspring are going to have the dominant and recessive versions inherited together. That's what linked genes means. Now, the person who figured out about linked genes is the same person who figured out about sex-linked genes, and that's Thomas Hunt Morgan. What he did with these experiments were a little bit different. In this case, he was looking at 2 genes, very similar to how Mendel looked at 2 genes for his dihybrid crosses. Also, just wanting to point out that Morgan used different letters for his allele names, and I know I mentioned this before, but it doesn't look quite right to our eyes. We're used to using big letters and little letters. Morgan used pluses and not pluses, like the lack of pluses, to indicate whether or not a trait was a wild type. And again, wild type just means most common. So it doesn't mean dominant and recessive. So in this case, we have a yellow body, and the yellow body is referred to by the BL plus allele name, because yellow is wild. And then if you have the BL without the plus, right, you just have that, then that would mean that you have the black allele. So this individual right here is heterozygous for body color, BL plus, BL, heterozygous. And then this VG plus, that stands for vestigial, and that vestigial is referring to wing shape. So you can either have normal or common wing shape, which is, I mean, the bigger wings. Of course, if you think about flies being able to fly, and flies can't fly with these tiny little vestigial wings. So, of course, the most common one would be the one where you could actually fly, so VG plus. This female, though, is heterozygous, so VG plus, and then VG, meaning she's heterozygous for body color. This male here, he is homozygous for vestigial, and he's homozygous for body color. So he's homozygous for wings, meaning and VG no plus, so that's the least common, so that means he has the vestigial wings. Again, I know this is a little confusing because so far we've been talking about dominant and recessive. But you can also figure out from this which traits are dominant and which ones are recessive, right? So remember that you have to be homozygous in order to show a recessive trait. So the vestigial must be recessive because this heterozygote shows the normal wings. And black body must also be recessive because this heterozygote shows the yellow body, right? So again, dominance and recessive does play a role here, but you can't just assume with Morgan's alleles, the way he names alleles, you can't just assume that that the wild type is dominant. You kind of have to figure that out, again, by looking at these things. So let's take a look at the same cross, but looking at it from where things are located on the chromosome. So what Morgan found was that body color and wing size are generally inherited together in specific combinations, whatever the parental type was. That's the type that you tend to get. So, so what he kind of really reasoned through is like if these had been on different chromosomes, then they would have assorted independently, but they didn't. So they must be on the same chromosome. So he also did notice that on occasion you would get the non-parental phenotypes. Let me just go through this idea of the parental phenotypes with you. So just kind of remember that the parental phenotype is related to whatever the parent was in this case. So we have a heterozygous female and we've got a homozygous recessive male. And so what you would expect would be to get, some random combination. It really should have come out as what we've seen with our normal independent assortment kind of concept. But that's not what was happening. Most offspring were either got this chromosome from mom, and of course one of them from dad. He only has the one kind to give. Or the offspring would get this combination from the mom, and of course the same one from the dad. With independent assortment, we would expect to see some of the wild type yellow bodies kind of being found with some of those vestigial wings. And the opposite should also be true if independent assortment was happening. We'd expect to see some of those black bodies found with the normal type of wings. But he just didn't find it, at least for the most part. There were some non-parental phenotypes produced, and there's your term right there, non-parental phenotypes. They were produced, but at much lower levels than were expected. But they did show up. And so what Morgan figured out from this is that you do get some sort of recombination occurring, genetic recombination that's happening at the level of the chromosome. So let's take a look at that. So again, looking at a very Mendelian kind of concept here, with some plays on the way that we're doing things. He took a wild type normal winged body. In this case, though, instead of doing a yellow body, he did a gray body. Don't ask me why. This got switched up. And then you can think about gray. It's grayish yellow. And then he did mate this female to a male that had a black body and vestigial wings. So when he did that cross, he took all of the females that were the F1 females. He didn't take the males, so stand by. And he mated those females to males that were showing the black body and vestigial wings. This is called an F1 dihybrid test cross. If you don't remember what the term test cross refers to, you can go back and look that up, but I'm just going to briefly remind you that test cross is when you're not sure of the genotype of one parent and you absolutely mate them to something that shows the homozygous recessive. So the reason that he was mating them to the homozygous recessive is because he wanted to control for the genetics of one of the parents and not pay attention to like having to deal with double crossing over and all of that other stuff. So the sperm that's contributing only recessive alleles, and the female is contributing some heterozygous combination of alleles. And when he did this, he got this very interesting result. The predicted ratio would have been 1 to 1 to 1 to 1, right? With females who were wild type, females who were vestigial and black, females who were wild type body but black with the vestigial wings, and ones that were vestigial wings, or sorry, normal wings and black body. So you would have expected to get that 1 to 1 to 1 to 1 ratio. But that's not what he got. He got 1 to 1. Well, let me just back up to here and say, you could, if those genes were completely linked, you would expect 1 to 1 to 0 to 0. But that's not what he got. He got something very interesting that is close to 10 to 10 to 2 to 2. It's close to that. Or maybe 9 to 9 to 2 to 2. Very close to that. And so that was very puzzling to Mendel, or to Morgan. It didn't meet either of the predictions that if they were on different chromosomes, 1 to 1 to 1 to 1, or if they were on the same chromosome and always linked 1 to 1 to 0 to 0. No, came up with something very strange. And so what we realize now is, of course, that everything's located on chromosomes, and chromosomes do recombine. Mendel, right, he figured out that combinations of traits of offspring do differ from their parent. And offspring with a phenotype that matches one of the parental phenotypes, that's called the parental type. The other one is called either the recombinant type, or we use the word recombinance. So, if you have a 50% frequency of recombination, and this is key here, this math right here is key. If you have a 50% frequency of recombination, then that means that the any two genes are on different chromosomes. They're going to be on different chromosomes. So that's cool. Like if we go back and think about our pea plants again, if you cross the gametes from dihybrid, heterozygous for both traits, round versus wrinkled, yellow versus green, and you do a test cross with the green, this one would be green and wrinkled, you would get a 1 to 1 to 1 to 1 ratio. And so that means that those are on different chromosomes. Lucky Mendel, he didn't have to deal with this concept of crossing over. But Morgan did discover that when two genes are on the same chromosome, some are going to recombine. And he proposed this concept of crossing over. Again, we know this concept of crossing over from dealing with it in meiosis. But Morgan is the one who figured out this concept of crossing over. So if you look at this cute little animation here, you can see crossing over happening. And notice that it doesn't happen at every single location on the chromosome. So any genes that are on this side are all going to be inherited together. But here's at this crossing over place, you're going to get some recombination. So in other words, recombinant chromosomes bring alleles together in these new combinations in the gametes. And again, this doesn't happen in you, it happens in your gametes. And so it's your offspring that get the new combinations. Random fertilization, remember, that was an important concept from before. Random fertilization also increases even further the number of combinations that can be produced. And so all this genetic variation, this is the raw material on which natural selection and evolution are going to be acting. And we'll get to be talking a lot about those in the final couple chapters of this semester. But we're not quite done, because there's a little bit of math associated with this. Now, I'm just going to be frank and tell you that I am not going to make you actually do the math, but I am going to ask you to understand the basics of this math, and I'm going to ask that you be able to apply the math, even if you're not going to be asked to actually calculate any math here. So this gentleman named Alfred Sturtevant, he was a student of Morgan's. he took all this crossing over information and he constructed something called a genetic map. Basically, all a genetic map is a list of where each gene is located on the chromosome. And what Sturtevant predicted was that the farther apart two genes are, the higher the probability of crossing over will be between them. And so therefore, you're going to have a higher recombinant frequency. So if you have a crossing over number that is low, if you have a genetic map where these two things are really, really close together on the chromosome, then you're not going to get a lot of recombination. If they're really, really, really far apart, however, you are going to get a lot of recombination. So with Morgan's results from before, you can see that there's some moderate amount of recombination. That means they're on the same chromosome, but some distance apart. What Sturtevant did was he made something besides just doing this chromosome map thing, right? The genetic map. He also did something called a linkage map, where he calculated the frequency of how often these two genes recombine. And so, and that's for whatever that frequency is, that's how far apart they were. So he did this in something called map units. So the distance between genes are map units, and one map unit represents 1% recombination frequency. So the distance between the body color allele and the wing shape allele, there was a 17% recombination rate, so they are 17 map units apart. And that's pretty much how it goes. Again, I'm not going to be making you do the math of this, just understanding it. Genes that are very far apart do tend to show approximately a 50% recombination rate. And so that means that they're linked, but they're not. There's a lot of crossing over that happens between them, or a lot of chances of having crossing over. But if you're closer together, you have fewer chances of getting a crossing over happening, or like right there in the center. And so you have lower amounts of those recombinant offspring. So he Sturtevant was able to map lots of different all the chromosomes in fruit flies using this technique, and looking at the crossing over rate. And so yeah, that's what you end up getting, is this, like, here's this gene, and here's this gene, and here's this gene, and so forth. He also used this concept of chromosomal banding, and banding is found on chromosomes, and that has really nothing to do with location of genes per se. But these bands show up whenever you look at karyotypes. And so geneticists were able to develop these things called cytogenetic maps. And basically, cytogenetic maps indicate where the positions of the genes are relative in kind of relative location to where these banding patterns are. So again, that's a cytogenetic map. And that's a little different from a gene map, although they're very, very similar. They're just like, oh, here's a gene, and it's located kind of near this stripe pattern. And here's another gene, and oh, it's located near this other striped pattern. So we were able to, I guess Sturtevant and Morgan were able to figure out where different genes are on these chromosomes. And here's just an example of what one of those maps looked like. So there's this eye color one here. There's wing shape with it to curved versus non-curved, and vestigial wing versus normal wing. Again, notice that we're not saying dominant and recessive here. Cinnabar eyes, those are very interesting stripes on the eyes. Body color, maroon versus red eyes, and antenna, appendages on the head, and so forth like that. Again, I'm not going to make you do this math. I just really want you to understand. And I'm not going to make you memorize where these different genes are on this chromosome either. Again, I just want you to understand that it is possible using these techniques to locate different genes within a chromosome. Well, that's all that I have for you for now on Morgan and Sturdivant's research on crossing over and linked genes and recombinant genes. We're going to be moving on to chromosomal aberrations, also known as chromosomal alterations, in the next video.